Here's part 1 of Chapter 8, Biology of Infectious Diseases. Some of this information may be redundant, at least at the beginning of the first few slides here from Chapter 7. Now, here's the best list for the Chapter 8 objectives. On this list, there are 2 additional objectives that are not in the list of Chapter 8 objectives that is posted directly in Canvas. But this list is a lot shorter than the list that is posted within the PowerPoint lecture notes. I was a bit ambitious just because that information is so interesting. And this is a really cool chapter because this is about microbiology, and that is my favorite class to teach. So, as you're going through the PowerPoint notes, the lecture or the audio material, have this list of objectives and start making a study guide for the next exam. Alright, so humans and microbes have a very close relationship. As I mentioned before, bacteria cells outnumber our human cells by quite a bit. We use microbes to help us make food, to degrade material in the environment. We wouldn't have life as we know it, or life at all, if it weren't for the bacteria in the microbes. So they are extremely important. As mentioned in Chapter 7, the microbes that cause disease in humans are called pathogens. So, microbes are the bacteria, the viruses, the prions, the viroids, fungus, parasites, and you have a nice assignment to go through, and that assignment is going to be material that will be used to make questions for the exam. Now, this slide is simply to show you a size comparison of comparing viruses to bacteria to eukaryotic cells, our cells. Viruses are generally much smaller than even the bacteria cells, the prokaryotic cell you see there on the bottom left, and both are much smaller than our eukaryotic cells, our cells. So, bacteria are single-celled. They are prokaryotic organisms. They are very simple, relatively simple compared to our cells. They almost all have a cell wall that helps for integrity and to keep their material safe. Their DNA is going to be found in a single chromosome. They do have ribosomes, but not everything else that our cells have. And some bacteria have what are called plasmids, and those are additional pieces of DNA that they have acquired over time. And usually bacteria are going to reproduce through binary fission, which is just the cell basically dividing itself in half. So you've got the binary 2, and it just cuts itself in half, the binary fission. And the 2 cells are going to be identical to the original cell. And we'll talk more about cell reproduction in humans, chapter 19, 20, 21, somewhere down the road. Alright, so bacteria have specific structures that are used to help it do its thing, whether that's being a pathogen or whatever the case may be. In this picture, in this figure, you see the plasmid, the circular piece of DNA. You have the cell wall here, but let's look at the things that may cause it to be pathogenic. Alright, these little hair-like projections are called fimbriae, hair-like bristles. This allows for a bacteria to attach to a surface. And if it can attach inside of our bodies, then it can stick there, replicate, and cause problems. Whereas if it just slides on through our system, we may never know it's been there. The capsule, this brown component here, is gel-like, it's sugary, and it will coat the entire surface, the exterior of the bacteria cell, and that actually causes it to be slippery to the white blood cells. And so they can avoid the immune system. And then some cells will have a flagellum, this little tail-like apparatus, and that allows it to swim, basically, to propel it through the environment. This can also help a pathogen or an organism be pathogenic. Now, additionally, bacteria can have different shapes, and this is a rod or a bacillus-shaped bacteria. These cells are circular, so they are cocci, and these are kind of club-shaped. These are spiral-shaped, and these shape features can actually be used to help diagnose an infection or what organism is causing an infection. All right, as I mentioned before, viruses are not alive. They are obligate parasites, meaning they are obliged to be inside another cell in order to do damage. So most of this information is repeat of what was mentioned in Chapter 7, so I won't go over it. We'll just skip on through to prions. Prions are interesting. They are infectious protein particles. They're not alive. They are simply protein. They don't have nucleic acid. It's just a protein. And these infectious particles cause damage within the brain. All right, the prion gets into the brain, causes the normal protein that is found in the brain to change their shape, and then all of those proteins eventually lose their shape. And if you remember from earlier, shape is important for proteins. And one form of prion disease is thought to occur when the normal protein can change all by itself, and the disease starts from that point. And these are the mad cow disease type infections. And in some cases, Kuru is the human disease, and I don't remember exactly how long ago this stopped, but the way that you can contract a prion is to actually ingest the prion material. All right, and with the human disease, Kuru, this was popular in New Guinea decades ago, and it was part of the tribal culture there. When a person died, that they would actually eat part of their brain. And then unfortunately, if the individual died from Kuru, then anybody who ate the brain of that person would then succumb to the disease eventually. So it doesn't sound appetizing, but it was simply part of their culture, and medical personnel or the World Health Organization personnel went there, figured out what was going on, and actually helped to change their culture, not because of what us Americans might think would be gross, but because it stopped the Kuru spread. All right, when we're talking about infectious diseases, we have a couple of terms. One is epidemiology, which is the study of how disease spreads in populations. Then there is infectious diseases. Those are diseases caused by pathogens. All right, diseases can be heart disease, which we've talked extensively about. That is not an infectious disease. All right, infectious disease is going to be caused by a pathogen, bacteria, viruses, prions, fungi. And then some diseases of interest that we're going to spend some time on are HIV and AIDS, tuberculosis, malaria, and influenza. All right. Just to give you a brief history of HIV, the origins are in Africa. HIV was originally found in non-human primates, and it's thought that it was first transmitted into humans back in somewhere probably in the 1930s, and it probably came into the human population several times from the chimps into the human population, probably through a butcher or a hunter that was unlucky enough to find a chimp that had this chimp HIV type virus, and then it mutated so that then it would do well, survive well in humans, and be able to pass on to the next person. All right, but the exact dates, it's unclear because it's hard to go back in time to take or to look at that. But they are, historians estimate that it was probably in the 1930s. The first documented death in the U.S. was in 1969, and this was, again, looking back, because it wasn't actually identified until the mid-1980s with the big outbreak in the San Francisco area primarily, but there were other hotspots in the United States as well. So, prevalence of HIV, the state is a little old, but approximately 34 million people are living with HIV worldwide. And not a good statistic, almost 1% of adults are living with HIV. And 1.6 million people died from HIV/AIDS in 2012. Thankfully, that number or the rate of death is declining, but it is known that over 36 million people have died from AIDS since the beginning of the epidemic. Now, most people living with HIV live in developing countries. So, the prevalence of HIV in North America, we have about 1.3 million people. In the Caribbean, huge number relative to the overall population there, 250,000 people. Latin America, 1.5 million. But look over here in sub-Saharan Africa, 25 million people. Just absolute devastating to that part of the world. All right, now this is one piece of information that you want for your study guide. What are the stages of HIV infection? All right, you have acute phase, you have the chronic phase, and then you have AIDS. So, looking at a timeline here, all right, years since infection. Right after infection, the purple line here is HIV in the plasma, so the amount of virus that can actually be detected in the blood. Very high initially. See, it peaks right up there. And then soon after infection, and I don't like this graph because it doesn't break that first year up into months or weeks, which is a little better, gives you a better time frame. But this time right in here, right, this high peak and this dip, probably looking at about 3 months on average. And so initially you have the virus load peak, and then it comes down. The reason for that is the virus is going to get into your blood and seek out CD4 positive T cells. These are the helper T cells. And if you remember, viruses only infect specific types of cells. They don't infect all types of cells. HIV infects CD4 positive T helper cells. And the virus peaks, and then what's going on here when it dips? It's because they're hiding out in these T lymphocytes. Some cells will die initially, and so the blue line here shows the number of helper T cells in the blood. You have normal levels here at the point of infection, and then it quickly drops. And the virus hides out in those T cells. And then over time, the first few years, the CD4 count after that initial drop declines a little bit, but stays about the same. HIV amount in blood relatively constant after that first year. And during this acute phase, in this initial phase, a person might just feel a little run down, but not necessarily know that they're sick. In this phase, they may not know that they're sick at all. Now, the chronic phase is when a person will start to get sick. And they're starting to get sick. Why? Because these helper T cells are starting to decline even further. And the helper T cells help the immune system to function properly. And without the proper functioning of the immune cells, you get sick. And then the final stage is AIDS, and that is when a person has virtually no helper T cells, their immune system clumps.